Any more question? I will happy if you will interact rather than pasting message in the chat box. I will be happy if you, any one of you interact. They can ask their query directly to me. Okay, though it is also a good platform.
So I think we should proceed for the practical session.
हाँ मैं प्रैक्टिकल स्टार्ट करता हूँ कपिल, मुझे लगा थोड़ा इंटरेक्ट कर लेते हैं तो ठीक है। चैट बॉक्स में कुछ क्वेश्चन थे, मुझे लग रहा है रिप्लाई कर दिए हैं, और अगर कुछ होगा तो फिर हम डिस्कस कर लेंगे, कोई इश्यू नहीं है।
ठीक
है ना, एक्चुअल में थोड़ा इंटरेक्शन होता है तो
वो अच्छा रहता है।
एक्चुअली वर्चुअल में यही प्रॉब्लम है, इवन जब हम क्लास भी लेते हैं तो कई बार ऐसा फील होता है कि हम ब्लैंक स्क्रीन को पढ़ा रहे हैं। सो इसलिए मुझे थोड़ा इंटरेक्शन करने का आदत है, दैट्स व्हाई आई एम रिक्वेस्टिंग एवरीवन, है ना। ठीक है, कोई इश्यू नहीं है। चैट के थ्रू
आशुतोष ने फिर से कुछ डाला कि ये सर बट
हाँ
प्लीज
आशुतोष का क्वेश्चन मैंने पढ़ लिया है, मैं उनको अभी आंसर वही कर रहा था एक्चुअल में। I will suggest better you use Tessel, because even a large HapMap file can be processed in the Tessel instead of using Mega for phylogenetic analysis. Even you can think of, I have analyzed 500, 600 genotype. I don't have exact idea. I have analyzed the genotypic data of 500 wheat lines with 35K chip, SNP chip. So I do not think you have data of 1,000 lines so that you can know using Tessel. So it is better you use Tessel if you have a file. So it will be easy to handle also. Okay. So if still you are not convinced, you can interact. We can interact over mail if you have any confusion or anything you want. If still you are not convinced, so we can interact, or even you can message me. There is no issue. Okay, so now what I have planned, because most of you are biologists, so you have gone through the very important resource called NCBI. So we will explore NCBI. We will see, somebody has asked what is gene expression omnibus. We will see all those things, how we can download the genome data, how can we download the assembly data, how can we get the sequence information, gene, everything we will explore using NCBI now. So just give me few. Are you able to see my screen? Or still you PPT is there? Are you able to see my screen? Google, I have opened in the web browser.
Not Google sir, still it is PPT sir.
PPT दिखाई दे रही है अभी?
हाँ अभी भी।
अच्छा ठीक है, मैं उसीलिए मैं पूछा था, एक मिनट मुझे शेयर करने दो, मैं फिर से शेयर करता हूँ, एक बार PPT अनशेयर कर लो। Just give me few minute time so that again I can share. I want to share the
Actually, there is some technical glitch, so Neeraj sir will join us soon.
Actually, there was some issue. My system become hang. I'm unable to share. So again, I am sharing the screen. Okay.
Okay,
Due
sir.
to that PPT, there was some issues. So again, I am sharing the screen. Now you can. You are able to see the screen.
Yes, sir.
Yes,
It
sir.
is
Yes, sir.
It is moving.
Visible.
Tab is moving.
Yes, sir. Yes, sir.
Okay, fine. So
Actually, it is stuck, I think.
yeah.
Stuck again, stuck, sir.
Oh, see.
No, sir, still no.
Don't know what is the issue.
Your video also paused, sir.
Same happens earlier
Mhm.
also, no?
Mhm. Hello.
I don't know what is the issue when I am sharing the Google screen. It is earlier there was also some problem in practical, or
No
it
sir,
was
no issue.
No,
it
sir.
कोई issue नहीं था.
नहीं सर, आप क्या इंटायर स्क्रीन डिस्प्ले कर रहे हो या विंडो डिस्प्ले कर रहे हो सर?
मैं तो इंटायर स्क्रीन कर रहा हूँ. शेयर स्क्रीन. स्क्रीन ही शेयर करनी है ना?
हाँ, उसमें एक इंटायर स्क्रीन है, एक ओनली विंडो है सर. तो
एक
इंटायर स्क्रीन पे करो सर, आई थिंक वो इजी है.
फाइल्स, एक मिनट. बेसिक मेरे पास जो आ रहा है स्क्रीन आ रहा है. लॉन्च कहीं आ नहीं रहा है इंटायर स्क्रीन.
सर मैं एक सेकंड में दिखाता हूँ. जो स्क्रीन आ रहा है ना, सिर्फ जो फर्स्ट वाला है ना सर ना,
हाँ,
ओनली स्क्रीन है, वो ही आपका इंटायर स्क्रीन है सर.
वही है ना?
हाँ, वही इंटायर स्क्रीन है.
ठीक है, ठीक है.
हम्म.
क्योंकि पहले तो कभी ऐसा कोई इशू नहीं हुआ.
हाँ, अभी ठीक है सर, अभी गूगल ऑन हुआ.
पहले ऐसा कोई इशू नहीं हुआ. अभी ठीक है.
अभी ओनली गूगल ओपन है सर.
कोई तो बड़ा इशू है. एनसीबीआई नहीं दिखाई दे रहा है.
नहीं सर, ऊपर खाली ओपन है, बट वो वैसे कुछ ओपन हुआ नहीं सर.
ऐसे क्यों हो रहा है?
सर आप आर के थ्रू कर रहे हो या कैसे जा रहे हो?
मैंने तो कुछ और प्लान किया था कि मैं जो है ये सब बताऊंगा.
मुझे अभी कोई इशू है स्क्रीन में?
नहीं, हार्ड खोल रहा है. एक बार क्रोम प्रोफाइल खोलना.
ओपन हो रहा है।
नहीं वर्क कर रहा है प्रॉपर्ली। आई थिंक सर के सिस्टम को कुछ इशू है। एनसीवी। इट्स वर्किंग प्रॉपर्ली। आई थिंक सर के सिस्टम का कुछ इशू
वो
है।
कर रहे चेंज कर रहे हैं आई थिंक सिस्टम।
हम्म।
हाँ सर।
जैसे ही आता है तो तुरंत क्लियर किया गया है। तो भाई इनके लिए बाहर नहीं डालना।
अभी तो काम कर रहा है सर ये। अभी प्रॉपर्ली वर्क कर रहा है सर। सर अभी वर्क कर रहा है, आप कंटिन्यू कर सकते हैं अभी तो। So please unmute yourself.
If still there is some issue in practical, so what I can do, one thing I can do, if you want to have any specific analysis, if I am unable to complete practical because there is some issue, though I will try to complete from my side. If there is any issue, suppose you want to do certain type of analysis like genome-wide associativity, genomic selection, phylogenetic analysis, any kind of a specific analysis. If I could not able to complete the practical, I will share you the code or some material. So don't worry. So this is technical issue. This is not in my hand and coordinator's hand. So such type of problem comes. So don't worry. If, even after practical, we are accessible. So you can always contact. So don't worry. Okay. So we have wasted 15-20 minutes. I don't know whether any issue still any issue arises. Don't worry. We can still we can interact, and if you want certain type of material, certain type of code, certain type of analysis, or any kind of that I will help. So don't worry about that. So what was my plan? What I have thought for you? I have thought that first we will see various kinds of information available in the NCBI. You can see if I have given a query of wheat heat stress. So you can see the availability of data like PubMed Central, then papers related to this, genes, GEO dataset, variance dataset, bioproject, biosample, nucleotide sequence, SRA sequence, number of data you can see related to heat stress. Somebody has asked GEO dataset, gene expression omnibus. So what comes under this repository is the gene expression omnibus data. It contains the expression data, that may be microarray, that may be RNA-seq data, high throughput sequencing data, chip-seq, single-seq, any kind of data you might find. Suppose I will start with that example only, that GEO dataset. So suppose it is related to wheat heat stress. So just click on GEO dataset. Okay. I hope it is moving.
Yes, sir. It is moving now. No issue.
Good. So you can see the availability of data set, series, samples, platform, number of things are there. If you want to restrict, suppose expression profile by array, methylation profile by array, suppose you want to restrict to certain type of sequencing platform, certain type of technology, so you can always select or customize accordingly. So you will get data related to that only. Okay. So this is called gene expression omnibus, where the data related to expression like transcriptome, microarray, RNA-seq, chip-seq, number of technology are there. So on the basis of that data is deposited to GEO dataset category. So we will see one example. We can see one example. Suppose I will give you one example. Acetic acid and treatment enhances the drought avoidance in cassava. Okay, so you can click on this study. There are 6 samples available. Expression profile by array. You can see the experimental detail. If your data is related to microarray, that is expression profile by array, there is one tool that is provided by NCBI, inbuilt facility to analyze such kind of data. So suppose we have 6 samples, you can just click on analyze with GEO2R. Okay, so you can also do analysis using this tool, but provided that condition is that if it is related to microarray, then only you can do the analysis using this utility. Suppose you want to do the single-seq RNA analysis, RNA-seq analysis, so for that certain tools and packages are there which are open source, that So that you can use. Suppose I will give you one example. There are two kind of condition is there. One is there acetic acid treated leaves, and another is water treated leaves. So you can see a clear cut two kind of groups. One is water treated leaves, one is acid treated leaves. So acid treated leaves you can consider as a treatment, and water treated leaves you can consider as mock or control. Suppose you define group. One is treatment, just give me 1 minute. There is some issue in this selection this groups. So just give me 1 minute time. So maybe I will give you the exposure what this tool does. There is some issue in selecting the group. So you can see there are certain options what you do, like what any type of adjustment of p-value, then transformation of data, apply the other kind of normalization on other kind of parameter, p-value, log fold change, then profile graph. If you will give this id gu extension, it will generate the view data for. Then you can see R script is also available. You can use this R script also directly in your R. It will work. So this is also one way of fetching the data. There is some issue. I am unable to make group. Otherwise, I could have given you the example. So this is R script. Suppose you have some data with you. You can see GSC ID. There are 6 samples in this. So you can see, and accordingly you can do this analysis using this R script. This R script will automatically fetch data from NCBI. You need to library certain you need to invoke certain library like GeoQuery, Lima, and UMap, and using this sequence you can fetch the sequence from NCBI directly. Okay, you can fetch the sequence from NCBI directly, and accordingly you can do the analysis. So suppose there are a number of things available at NCBI. Suppose you want to see the genome data, just do wheat. You want to see the or specific triticum aestivum. So you can see My screen is moving, Kapil?
Yes, sir. Yes, sir.
अभी ठीक है ना?
हाँ, ठीक है, sir.
Okay, okay. So you can see the data. Like suppose I want to see the whether it's genome assembly or genome information is available or not. So you can see assembly, number of sequence assembly are there, which is available for wheat. So in this way, suppose I will give you one example. Assembly. You can see, just see IWGSC RefSec version 2.1. You need to only have finished genome. Critica maximum version, different version, IGSC version. So in this way, you can download the different assembly. Suppose you want to see the genome. Search in this way. You can see directly all annotated genome. You can see all information. In this way, you can download. You can click. So in this way, you can see various finished genome. It is a .fna file, so you can directly look down. Then you can use this with transcriptome analysis, genome assembly analysis as a reference set. Okay, so this is now you all know a very popular NCBI utility that is called BLAST. That is basic local alignment search tool. So you can see there are various versions of BLAST, like nucleotide BLAST, then BLAST S, T BLAST N, protein BLAST, various kind of BLAST, and then you can see some nowadays some other advanced version like Smart BLAST, Primer BLAST, Mega BLAST, Model BLAST. Number of BLAST version is there, advanced version, so that you can explore on the basis of your need. Suppose you want to do nucleotide BLAST or protein BLAST. Protein BLAST. So you can use certain sequence like any FASTA sequence. Suppose I am using one sequence, one example sequence. Then it will search again certain database, NR database, BLAST-P. You can have some other advanced parameter like algorithm parameters. So if you want to customize, otherwise, if you do not want to very much specific, you can leave the thing as such. So in this way, you can utilize all the NCBI utility like BLAST, like other utility GEO2R, all these things you can utilize. So it took certain time. it took certain time. So this BLAST is mainly used to search the similarity of our sequence. Suppose you find any sequence, you do not have any sequence annotation of this sequence, so better you BLAST. This BLAST term is so famous that now people use it in place of searching. They use do BLAST. This term is so famous among the bioinformatician or biologists. They directly say that you do BLAST. So doing BLAST means it is searching your sequence, your homology of your sequence, similarity of your sequence against the sequence available in the public domain. Okay, so how you can these restrict that sequences on the basis of e-value, on the basis of p-value, on the basis of query cover, on the basis of total accumulated score. So there are certain criteria, although NCBI already give you the information in sorted way. The important what we say, that important relative or close relative to the distance relative. You can see the example, how it has given information. You can see the query cover. Query cover is 100%. E value 0.00 means, or less than means, that is it is very less. It means this is good for us. E value smaller means there are less number of chance, less number of expected sequence in the database that has similarity by chance. I hope you already know that what is the difference between homology and similarity. Homology is the statement about some kind of evolutionary divergence over time, and similarity is just matching of sequence, nothing else. Okay, so you can see the percentage identity, query cover, then length, then score. So on the basis of that, you can say our sequence is similar to this sequence has corresponding accession, and all these detail you can accordingly find out. Okay, so this is one of the most commonly used tool, that is called BLAST. So there are number of things. It is just like a sea. Number of things, number of public available sequence is available in NCBI. It is depends on you how you mine this NCBI database. Okay, suppose there is one more very popular tool that is called ensemble plan, which is very popular. I will just give you overview. What is that? कपिल स्क्रीन मूव हो रहा है ना, कोई इशू तो नहीं है अभी?
नहीं सर, कोई इशू नहीं है सर.
Fine. So you can see, you can select the genome provided that it is completely sequenced or whatever state it is. If it is available in the public domain, it must be here. It will be here only. So you can see the Arabidopsis or Ija, Triticum, Hordia, Giamage, then other genome. So you can see, suppose I am taking the example of Triticum estigma. It will give all the information like genome assembly, IWGST genome assembly, its FOSTA sequence, then convert data to IWGST coordinate, then other GFF file, other annotation file, phylogenetic overview of gene family, alignment, all the information related to that genome. One more thing you can also do. If you go to this option, download, you can directly see like Arabidopsis thaliana species, this FASTA sequence, cDNA FASTA sequence, CDS sequence, non-coding RNA sequence, protein sequence, annotated sequence to gene bank, and a gene set, any other number of information is available. Number of variation information, you can see VCF information. Number of information are available. Okay, so you can explore all these things. It is a very good resource, ensemble plan. Okay. like that. GFF file, FASTA file, all annotation file, everything GTF file. This you all can get from here. So this is also a very good resource for biologists. Then one more thing that is called most popular for protein, that is called PDB, Protein Data Bank. So you can see, you want to download the coordinate, you want to download the structure, everything. All the beauty of this PDB is that it has the protein structure available, which are solved through X-ray crystallography or NMR. Means they are wet lab data. Actually, it is not in silico, it is wet lab data, solved protein structure solved through wet lab technology. You can see the popularity of this resource, that to date it has 211,377 protein structure sequence of protein structure is available in the public domain. So you can see anyone which is latest entry. You can click on the latest entry. You will find all detail PDB file, all its experimental detail, all other parameter. You can see display download file. You can download FASTA sequence, PDB format, and number of formats are there. So in this way you can see. So this is also a very important resource, PDB. So now we will come to one important analysis, that is called what we call this phylogenetic analysis. So there is one tool called MEGA. कपिल अभी स्क्रीन मूव हो रहा है
सर, हो रहा है मूव है सर।
मेगा ये दिखाई दे रहा है क्या
हाँ
स्क्रीन
हाँ
पे?
हाँ सर, चल रहा है चल।
ठीक है, तो मैं मेगा में कुछ कराना चाह रहा हूँ, तो क्या डाउनलोड कराऊं सब लोगों को?
नहीं सर, बस खाली आप एक्सप्लेन कर दीजिए इसको
हाँ,
कि कैसे
वैसे यहाँ
करना है।
पर हाँ मोस्ट ऑफ द लोग ऑलरेडी इसको यूज भी कर रहे होंगे, बहुत सारे लोग यूज भी कर रहे होंगे, तो दे ऑलरेडी नो। और मैं कुछ ट्यूटोरियल भी शेयर कर दूंगा, कुछ अच्छे ट्यूटोरियल भी शेयर कर दूंगा, अगर किसी को एनालिसिस में कोई इश्यू आता है या कुछ भी आता है, सो दे कैन यूज दैट। ओके, so this is home page of MEGA, molecular evolutionary genetic analysis. So it starts with basically the basis of any phylogenetic analysis. Even any advanced, any basic to advanced analysis is sequence alignment. Okay, so first it starts with sequence alignment only. So I have one sequence file, MEGA file, FASTA file, analyze or align file, align. You can see align using cluster W. Most of you are already aware there is one tool that is used for multiple sequence alignment. The basic recipe of any phylogenetic analysis is multiple sequence alignment only. So before building any phylogenetic tree, better first we will do the starting step is that to do the multiple sequence alignment. So you can see there are 10 sequence: human, cat, cow, buffalo, chimpanzee. They are belongs to a particular gene, belongs to various species. Okay, so how you can do that analysis alignment? Align by, there are number of method. Cluster W, muscle, it depends on based on their varying scoring strategy, then their strategy how they align the sequence. So there is, it is not like that there is a drastic difference between the cluster W, muscle or different tool. There may be difference in their how they are parallelizing gap, how they are giving the score match, mismatch, or maybe gap opening penalty, gap extension penalty. So the algorithm may differ in certain parameters. That's why some result may vary. Otherwise, more or less the results are similar. So if I use align by cluster W, so first you need to select all. Okay, so there are certain parameter: gap opening penalty, gap extension penalty, then other scoring scheme. So it is better you run by default. If you have a very good idea of all these, so you can change the penalty also, or the scoring scheme also. Okay, so this you can see the alignment. What happens? What algorithm does? Algorithm try to match column wise position of particular sequence throughout all the sequence. So whenever it is possible, it try to match. Otherwise, it insert gap, or it may also align mismatch. So this is the way how this multiple sequence alignment algorithm works. So now this alignment is basic recipe for phylogenetic analysis. Suppose data, you can save this session, or you can export this alignment. You can export this alignment mega format like that. Just save this and you can import this. Now I will import. Just see data, saved alignment session. Just give me file, open a file or session. Now I will open that align one. That was FASTA, which I have earlier did. Now it is aligned. So just give me 1 minute. Okay. You can see phylogenetic analysis. You can choose the statistical method, maximum likelihood test for phylogeny, any bootstrap method. Bootstrap method is the important method. So this you can do to cross verification whether that it is placed on the right node or not. So in this way you can do. There are certain other parameters. For that you need to have some basic knowledge of these parameters. It is not like that. Do not use these parameters randomly. You must have some idea about all these information. So if you do this, so it may take some time. Because why it is taking time? Because we have chosen the bootstrap method. Otherwise, if you have not chosen bootstrap method, it may be analyzed very quickly. It will take some time. I want to abort it. I will give you some other example. Suppose it is better not to use bootstrap. Okay, so now you can see pre-generated. So there are certain things you can change the orientation, you can see the beautification, how it will look like. Then several other, you can increase font, you can color. There are a number of things which you can do here. You can export this into particular format. You can create some other, you can create the alignment from here, and you can save this alignment information into some file, some format, and you can use this imported, like New Week, Nexus. There are a number of phylogenetic or alignment format are there that you can use in other tool also. So like that, you can use this type of information. So then you can use, there are a number of things. Then compute amino acid composition. You can see how composition of amino acid is there, which amino acid is there. Like that, number of downstream analysis are possible. You can explore diversity. You can also do number of things you can do from here. So this is a very important and comprehensive tool to do such kind of, and you can construct UPGMA. There are number of methods: minimum evolution range, maximum likelihood, neighbor joining. The most commonly used among the scientists is neighbor joining. It is very important, neighbor joining and UPGMA. Okay, so in this way you can use these tools. Or it is also used for multiple sequence alignment. You need not to go to use cluster W directly. That utility is available in Mega also. So you can use that Mega tool also to do the multiple sequence alignment. So this is all about this Mega. You can download. Most of you have already have this tool, so you can use this for multiple sequence alignment, then phylogenetic analysis, or other kind of evolutionary analysis like diversity analysis or all these things. Okay, so any specific thing you want to discuss with me? Actually, this topic was very broad, so it was very difficult for me to focus on certain things. That's why I have to have a broad idea of practical. Otherwise, if it may be related to particular specific study, then we can have a very streamlined practical, since it is a very broad topic. So I thought better to make you acquainted with major resource available on the public domain, how you can use those resources, how you can use this type of web utility available in NCBI for analyzing your data. So I think this is all about the practical part. Still, if you have any specific issue like analyzing intranscriptome data or handling QTL analysis data, GWAS, genome-wide association study, or you can say genomic prediction or genomic selection study. So you can ask for any help if you have any issue dealing with these analysis. Okay. So we can interact. Any one of you want to ask something or discuss something? It is not like that we are associated to this lecture only. We can have future collaboration. We can have any kind of help you want from our side. It may not be always in the form of publication. It may be other kind of help also. Sometimes you may be stuck somewhere in some analysis. So you can always ask. There is no issue.
Hello,
about
sir.
that. Yeah.
Sir, I want to know about. I want to download whole gene family through NCBI for heat gene expression profiling against heat stress. Sir, how can I do it?
What you want to download?
Sir, whole gene family to design the primer, sir, for gene expression profiling against heat stress.
Whole gene family. So
Yes, sir.
Which family you want to download?
Sir, heat shock gene.
Okay. Maybe in NCBI directly there is no option to customize the such kind of information if you have search in NCBI. When you
Sir, one by one, it will took more time.
how many such type of gene are available?
Uh,
Then I will suggest, if suppose it is 20, 40, 50, then there is no issue. You can just check box and you can check the sequence and you can download in a single file. Suppose it is in 500,000, so then you need to take the help of Perl scripting all these things. That's why I am asking the volume of data. Suppose if it is directly, if it is 40, 50 sequence, then there directly you can check the box and you can download. But suppose if it is in 1,000 or 500 like that, so then you need to take help of Perl or BioPerl. Then only you can easily download. So that we need to see what is the volume of information, because sometimes it happens, suppose we want to download, suppose you want to download 2,000 3,000 sequence, so generally we use BioPerl scripting, so that we need to customize that. For that we need to give some time. We need to what is our problem, that we have to do that scripting, then only directly you can fetch the data from there.
okay, sir.
Okay.
Okay, sir. Thank you, sir.
Okay.
विजय
So thank you so much, Neeraj sir, for this wonderful lecture.
थोड़ा बीच में डिस्टरबेंस हो गया, एक्चुअली ऐसा होता नहीं है कभी, पता नहीं। आई एम
तो
आल्सो
सर कभी कभी वो
सरप्राइज्ड।
टेक्निकल ग्लिच है, तो इंटरनेट का भी इशू हो सकता है सर। पर ये था कि मतलब बहुत सारी चीजें कवर करने का था, बट हमारे पास
नहीं,
टाइम बहुत ज्यादा
एक्चुअल में क्या है
लिमिटेड
ना,
था, इसलिए
मैं अगर ज्यादा
Yes,
खुश होता ना, अगर टॉपिक थोड़ा स्पेसिफिक होता, लाइक जीनोमाइड एसोसिएशन लाइक क्यूटीएल मैपिंग या
हाँ,
जीनोमिक सिलेक्शन,
जी
तो
जी
मैं
सर।
ज्यादा अच्छे से, क्योंकि जब वास्ट हो जाता है ना, तो
हाँ
चीजों
हाँ
को थोड़ा नैरो डाउन करना बड़ा मुश्किल हो जाता है, इसलिए मैंने
बिल्कुल।
ब्रॉड ओवरव्यू दिया, क्योंकि ऐसे प्रैक्टिकल कराना बड़ा मुश्किल हो जाता है हमारे लिए,
नहीं सर,
है ना।
एक्चुअली ये बहुत बेसिक चीजें थी,
हाँ
ये जानना
बेसिकली,
जरूरी था, इसीलिए हमने जनरल जैसा रखा था बायोइन्फॉर्मेटिक्स का,
हाँ
जिससे क्या है थोड़ी थोड़ी चीजें सबको एक आइडिया
थोड़ी
लगेगी
थोड़ी
किस
चीजें,
तरह से
हाँ
चीजें वर्क करते हैं।
हाँ वही।
So thank you so much, Neeraj sir, this wonderful lecture, and definitely our participants will means gain benefit from this lecture. So sir, आप अपना थोड़ा सा ये स्क्रीन शेयर ऑफ करेंगे सर एक
अच्छा
बार।
एक, ओ सॉरी, हाँ मैं भूल गया,
हाँ।
श्योर हो गया।
हाँ सर
हाँ
हो
हाँ
गया।
सर, 1 सेकंड।
एक मिनट, ओहो मैं तो इसी से परेशान हूँ। फिर से हैंग हो गया मेरी साइड से। ओहो।
सर आपकी आवाज तो आ रही है, बट आपका वीडियो फिर से पॉज हो गया। सर चले गए क्या?
सर ज्वाइन कर लेंगे अभी, ठीक है।
सो एक्चुअली पार्टिसिपेंट्स, हम चाहेंगे कि आप अपना एक बार वीडियो टर्न ऑन करें, जिससे कि हम स्क्रीनशॉट ले सकें आपका। तो प्लीज टर्न ऑन योर। सो अभी तक पूरे ट्रेनिंग सेक्शन में हमें मतलब बिना फेस देखे हुए सबको ट्रेनिंग करवा रहे थे। तो ये फाइनल लेक्चर था जो अभी कंप्लीट हुआ है। तो मैं चाहूंगा कि आप लोग अपना फीडबैक जो भी देना चाहते हैं आप दें, किस तरह से हम ट्रेनिंग को और इम्प्रूव कर सकते हैं और दूसरी चीजों के लिए वर्क कर सकते हैं। तो आप जो भी देना चाहे आप बोल सकते हो।
हेलो सर, गुड आफ्टरनून सर।
हाँ, गुड आफ्टरनून नासरुद्दीन।
हाँ सर, ऑडिबल हूँ मैं?
या
हाँ
यू आर ऑडिबल।
हाँ सर, तो सर मैं बोलना चाहूँगा बहुत ही थैंकफुल हैं हम सब सभी जितने भी पार्टिसिपेंट्स हैं, क्योंकि इतना कॉम्प्रिहेंसिव तरीके से कोई भी ट्रेनिंग शायद ही ऑर्गेनाइज किया गया होगा, मतलब जिसमें कि हम क्यूटीएल मैपिंग, जीवास मार्कर सिलेक्शन, जीनोमिक सिलेक्शन, बायोइन्फॉर्मेटिक्स सब एक ही ट्रेनिंग में हमने, जितना हमने 1 साल में सीखा 2 साल में पीएचडी 5 दिन में कवर कर दिया आपने। तो रियली वी आर ऑल ग्रेटफुल टू ऑल ऑफ यू, स्टार्टिंग फ्रॉम सुभाष सर ने पहले दिन में जो समझाया हमें बेसिक जो बायोमेट्रिक्स मतलब बिल्डिंग का एनालिसिस होता है, भरत आलम सर ने, फिर अभिषेक सर, अमरकांत सर, आज नीरज सर ने बहुत ही बढ़िया तरीके से ऑर्गेनाइज किया गया है। तो स्पेशल थैंक्स टू यू और कपिल सर, दोनों ही बहुत ही अच्छे से इसको प्लान किए। तो आगे भी हम चाहेंगे कि जो भी हमारे क्वारीज हैं, एक्चुअली हम चाहेंगे कि कुछ हैंड्स ऑन ट्रेनिंग हो, क्योंकि ये भी बहुत ही अच्छा था, जूम में भी होके भी इतना बढ़िया गया। तो अगर हैंड्स ऑन होगा तो हम ज़्यादा इंटरेक्ट कर पाएंगे। तो रियली वी आर ऑल थैंकफुल टू यू ऑल, थैंक यू सो मच।
थैंक यू
तो फिर
आशुतोष।
ऑफलाइन ट्रेनिंग
एक्चुअली
कराइए आप लोग। एक्चुअली हम लोग भी जब ट्रेनिंग कराते हैं तो हमारे
हाँ
वहाँ
हाँ
यही
सर।
ये सबसे बड़ा मतलब एक ये होता है, क्योंकि हमें भी फील होता है, दिस वी ऑल्सो फील इफ वी आर नॉट एबल टू डू हैंड्स ऑन वेरी, हमें भी ये चीज फील होता है। सो क्योंकि हम लोग भी ट्रेनिंग कराते रहते हैं ISRI में, तो जब भी वर्चुअल ट्रेनिंग होती है तो एक हैंड्स ऑन वाला थोड़ा इश्यू आता है।
सर एक्चुअली ये
हाँ
ऑनलाइन के भी इतना बढ़िया
हाँ,
हैंड्स ऑन, मतलब
ये थोड़ा
प्रैक्टिकली
इश्यू आता है। हाँ
ट्यूटोरियल मैपिंग जीवा कल समझा
हाँ,
रहे थे, तो प्रैक्टिकली इतना अच्छे से बताया कि ये
हम्म,
फील कि हैंड्स ऑन होता तो और, बट स्टिल हैंड्स ऑन अपना।
बिल्कुल बिल्कुल। अब एक्चुअली क्या होता है सर, जैसे अगर हम हैंड्स ऑन में अगर
हाँ
देखते हैं तो मतलब बहुत लिमिटेड पार्टिसिपेंट्स हो जाते हैं।
हाँ, फिर वो
और
इश्यू
दूसरे,
यही है ना कि हाँ अब
यस सर,
हाँ, जैसे अभी आपने 200 पार्टिसिपेंट को आप लोग ये कर रहे, तो सब 15 20 25 50 से ज्यादा तो हम कर नहीं सकते, क्योंकि उसमें
यस सर,
लॉजिस्टिक और भी इश्यू आते हैं। सो वो तो एक इश्यू है ही। अब वर्चुअल का एडवांटेज है तो डिसएडवांटेज अपना लिमिटेशन भी है।
लेकिन
यस
सर
सर।
इसका ज्यादा ऑडियंस तक पहुंच पाएगा क्योंकि यूट्यूब में
हाँ
अवेलेबल बहुत
हाँ,
सारे लोग हैं,
वो
तो
है हाँ।
और कोई अदर अगर कैंडिडेट या पार्टिसिपेंट अगर बोलना चाहते हैं?
चलो
कोई भी अपना
पार्टिसिपेंट
फीडबैक।
चैट बॉक्स में लिखना पसंद कर रहे हैं
आप
सब।
बताओ, कुछ बताओ हमें कि कैसे और हम इम्प्रूव कर सकते हैं, एटलीस्ट आप बताइए तो सही।
कोई भी बोल सकता है अच्छे से, जो
यूट्यूब।
भी हमारी कमियां लगी हैं आपको कि इनको इम्प्रूव करना चाहिए, तो सारी चीजें आप बोल सकते हो।
It is not like that. We also want to learn and improve.
यस।
हाँ,
It's not like that. That we
वो
will
नहीं।
take on future. We will
रसपाल
take
सिंह
care.
इज देयर?
रसपाल।
हाँ, रसपाल आई थिंक।
हेलो, नमस्ते सर।
नमस्कार।
हाँ जी सर, बहुत अच्छा लगा सर ट्रेनिंग। और जैसा कि सभी ने बताया सर कि अगर हम ऑफलाइन इसको ऑर्गेनाइज कर पाए आगे जो भी ट्रेनिंग हो, तो वो हमारे लिए ज़्यादा हेल्पफुल रहेगा सर। और इसके साथ साथ सर अगर जो भी हमारे आपने लेक्चर्स सर लोगों ने दिए हैं उनकी अगर मेल वगैरह वो हमें प्रोवाइड करवा दिए जाएं सर, तो उससे रिलेटेड जो इन्फॉर्मेशन है सर हम फर्दर उनसे कांटेक्ट कर सकते हैं। तो सर ये हमारे लिए थोड़ा सा बेनिफिट रहेगा सर।
No issue. आप लोगों को PPT मेल सब कुछ प्रोवाइड करा देंगे आपके ऑर्गेनाइजर। So that
हाँ
is
जी
not
सर।
the issue.
और कोई
सर एक्चुअली
कॉन्टैक्ट
हम अभी
सर,
एक
चाहे
ट्रेनिंग
मेल के
मैनुअल भी
थ्रू
प्रिपेयर
चाहे
करेंगे, तो
कुछ
आपके
भी सर।
डिटेल्स वगैरह इनके साथ में भी हम शेयर कर देंगे सर।
No issue.
जैसे कि
हाँ जी
इनको
सर।
कभी कोई क्वेरी होती है तो आपसे डायरेक्टली भी कॉल कर सकते हैं। और वैसे ये जैसे ट्रेनिंग मैनुअल रहेगा तो उसमें भी सारी चीजें जो मे भी छूट गई हैं वो सारी चीजें भी हम इंक्लूड करेंगे सर।
हाँ जी सर, थैंक यू सर।
ओके। और अभी ऑनलाइन कौन है? दिव्या एस, अनुज मिश्रा, कौन है?
सलमा सलमा इज देयर ना?
नहीं, जस्ट मैं उन्हीं को कॉल कर रहा हूँ जो यूट्यूब उनका वीडियो ऑन है, उन्हीं को कॉल कर रहा हूँ रैंडमली।
हाँ हाँ, सलमा यू कैन ऑल्सो शेयर योर व्यूज।
Yes, sir. Sir, it was very informative workshop, and we could clear so many doubts in that. Doubt about the different softwares and all. It was really helpful, and I think it will be very helpful for doing our PhD when we come across these statistical techniques. So I'm very grateful for this workshop.
थैंक यू वेरी मच। एनीवन एल्स लाइक दिव्या, अनुज, एनीवन?
Sir, may I?
हाँ
हाँ,
यस यस।
Hello, sir. I am Akhil Donga from Sardar Krishinagar Datiwada Agriculture University, गुजरात। सर, एक्चुअल में जो जीवास के ऊपर जो टॉपिक जो सेशन हुआ वो एक्चुअल में बहुत ज़्यादा इंफॉर्मेटिव था। उसमें से बहुत कुछ सीखने को मिला। जो डाउट्स थे, क्वेरीज थी, वो क्लियरली सर ने वो एक्सप्लेन की। और सर बाकी तो सब जैसे बोल रहे हैं कि अगर ऑफलाइन ट्रेनिंग या हैंड्स ऑन ट्रेनिंग हो तो वहाँ पे भी हम अपने जैसे कि सर वहाँ पे एक्सप्लेन कर रहे हैं और साथ में हम भी अपने पर्सनल पीसीस के साथ वो प्रैक्टिकल रन आउट करें तो ज्यादा अच्छा रहेगा।
बिल्कुल बिल्कुल।
हम करवा रहे हैं, जल्दी करवाएंगे, आपको इन्फॉर्म।
ऑफलाइन का भी हम ये प्लान कर रहे हैं। एक्चुअली हमारे वाइस चांसलर भी हमें यही चीज उस चीज के लिए कर रहे हैं, तो हम प्लान कर रहे हैं, तो जितने भी रिसोर्स पर्सन होंगे हम सब उनको बुलाएंगे साथ में, तो आपके लिए हम ये प्लान करेंगे, जरूर करेंगे इस चीज को।
थैंक यू सर।
विजय ठीक है, बैठ
तो कोई और कुछ बोलना चाहेगा? So this this was our last training session delivered by Dr. Neeraj sir. Thank you, Neeraj sir,
Welcome you all.
for joining us, sir, and for your presentation, sir. So after this completion of this session, we will provide you a feedback form. After that, we will provide you certificates. So maybe in coming week, we will distribute all the certificates to the participants. So thank you so much, all of you, for joining us and making this event very wonderful. And thank you, Neeraj sir.
Thank you all. Thank you,
Thank you.
Kapil. Thank you.
Thank you, sir.
Okay, thank you everyone.